Dear students, in the last lecture we had studied three models of the plasma membrane. One was fluid mosaic model given by SINGH and Nicholson, and another was unit membrane model by Robertson, and then sandwich model by Denali and Davison. And the acceptable model, as I told you, was the fluid mosaic model, and which explains the transport of materials inside the cell and outside the cell through the plasma membrane. It is because of this fluidity the solutes and could move in and out through the plasma membrane. Those molecules which are easily soluble in the lipid bilayer, they could move through the plasma membrane easily, whereas those which are polar in nature, they require special proteins called as porin, which lets in them inside the intracellular fluid. The molecules of the plasma membranes, they are in translational motion because of the weak interactions they are having. The weak interactions are ionic and hydrogen bonds. The plasma membrane largely contains proteins and lipids. The proteins are intrinsic as well as extrinsic proteins. They have various functions to perform in the plasma membrane. Both proteins and lipids, they are amphipathic in nature. That means they have hydrophilic as well as hydrophobic part. The polar ends or hydrophilic ends of the lipid bilayer remains inclined towards either ECF or ICF, whereas the fatty acid molecules, they remain oriented towards the middle portion of the plasma membrane, which is hydrophobic in nature. Now the proteins, they are also of having two kinds of domain. One is hydrophilic domain and another is hydrophobic domain. The hydrophilic domains, they remain in outside the plasma membrane, facing towards ECF or ICF. The hydrophobic domain of the protein is made up of those amino acids which are non-polar in nature. This hydrophobic domain or amphipathic nature of the protein has a great importance in the sense that they could be integrated in the non-polar lipid bilayer. So this was the introduction to what we had studied last time. Then this in the fluid mosaic model, then this was the unit membrane concept, and it says that the bimolecular lipid layer is sandwiched between two protein layers, but it hasn't been accepted. Lipid protein, which is the sandwich model, it was given by Denali and Davison in 1935. Then we were studying membrane proteins. Membrane proteins were of two kinds: integral proteins. Integral proteins, as I have mentioned right now, they are have hydrophilic domain and hydrophobic domain. The hydrophilic domain contains polar amino acids, and the hydrophobic domain contains non-polar amino acids and remains embedded in the bilayer, forming hydrophobic interactions with the fatty acyl chain. And I had mentioned some of the proteins which are integral proteins. This might come in your examination, that the integral proteins are either porins, cadarins, integrins, selectins, band 3 proteins, glycoproteins, sialoglycophorins, and some antigenic determinants which is also called as glycocalyx. And the organ acceptance and rejections is largely depend upon the antigenic determinants which are present on the plasma membrane of the organs or the blood. Integral proteins penetrate the lipid bilayer and have domains that protrude to both cytoplasmic side, that is ECF and ICF, and they are involved in such a strong interaction with the non-polar part of the lipid bilayer is that it is very difficult to separate them from the plasma membrane. It is because of hydrophobic interactions with the non-polar region with the fatty acyl chain. Now we can classify these integral membrane proteins into monotopic, bitopic and polytopic. The monotopic integral proteins are embedded in lipid bilayer exposed at only one membrane surface. The examples are cytochrome C and B5. Bitopic are the integral membrane proteins in which one transmembrane segment exposed on both the sides, and the examples are plasma membrane receptors, growth factors and certain peptide hormones. Then polytopic, more than one transmembrane segment weaving back and forth, displayed by adrogenic receptors and receptor particles of endoplasmic reticulum. Then certain polypeptides in bacterial and bacteria. Then we have another kind of membrane proteins which are called as peripheral membrane proteins, and they are not embedded in the lipid bilayer. They are located entirely outside the bilayer on extracellular or the cytoplasmic or ICF site. Yet they are associated with the surface of the membrane by non-covalent bonds. I have mentioned you in this lecture that the interaction between proteins and lipids is largely ionic, but the peripheral membrane proteins, they remain attached to the lipid molecules by non-covalent bonds. The examples are spectrins, actins, ankyrin, catenins. Then we have glyceraldehyde 3-phosphate dehydrogenase, which are non-covalently bound to the polar heads of the lipid or integral proteins. You can see in the slide that at the surface you can see black colored proteins. These are peripheral membrane proteins, whereas the red one having a coiled portion is the integral membrane proteins. The peripheral membrane proteins, they don't have two domains, that is hydrophilic and hydrophobic. They have only one kind of domain. These peripheral membrane proteins have a function to perform, and they provide mechanical support to the membrane during the rapid changes in the cell shape. Now there are another kinds of proteins which are called as lipid anchored, located outside bilayer but covalently linked to the lipid moiety situated inside bilayer. This is the difference between peripheral membrane proteins and lipid anchored. Lipid anchored proteins, they are anchored covalently to the lipid bilayers, which is lipid moiety which is situated inside the bilayer, and these bound to membrane by short oligosaccharide linkers. The examples are glycophosphatidol, inositol on the outer side. And lastly, we have lipids also, and they can be categorized into three: phospholipids, glycolipids and sterols. The polar lipid contains hydrophilic heads and hydrophobic tails. As I've told you in the last lecture, that the hydrophobic part is constituted by the fatty acids, and fatty acids could be saturated or unsaturated. These hydrophilic and hydrophobic tails, they are bridged or bridged by glycerol moiety. The phospholipids is one category of lipids, membrane lipids, and among the phospholipids we have glycerophospholipids and sphingophospholipids. Then we have another category called as glycolipids. Cerebrocytes and gangliosides are glycolipids. Then we have sterols, and sterols are cholesterol, phytosterol, and ergosterol. They are also have some functions to perform, which we'll study in detail in the upcoming classes. So students, this is all in this lecture. In the upcoming lecture, we'll study about the functions which are performed by the plasma membrane. Thank you so much.